In deep-sea footage, a pale squid hangs upright, while thread-like arms extend far beyond its body. Proportions look mechanically impossible, as though an ordinary squid were stretched without breaking. Yet several recorded animals share this architecture, identifying a real group rather than a camera illusion. Bigfin squids belong to the family Magnapinnidae, known from specimens and a limited number of observations. Their enormous fins, compact bodies, and extremely long arm filaments create the unsettling silhouette. The mystery is not whether they exist, but how that shape functions in deep water. Most filmed individuals appear briefly beside remotely operated vehicles, illuminated against an otherwise black landscape. Distance, angle, current, and incomplete views make exact dimensions difficult to recover from many encounters. The longest visible structures are slender distal filaments, extending from thicker arm and tentacle bases. They often bend at sharp-looking elbows, where the thicker portions transition into trailing filaments. That posture violates no anatomy, although it contradicts familiar images of fast, streamlined coastal squid. If you meet the animal only as a silhouette, scale and orientation can become surprisingly uncertain. Verified specimens are mostly small or immature, while some filmed animals appear considerably larger. This gap between preserved bodies and remote imagery drives the central investigation. The apparent impossibility dissolves only when form, water, behavior, and evidence are examined together. Magnapinnidae specimens possess large terminal fins that may approach or exceed the mantle's width. Their arms and tentacles show similar external proportions, unlike the obvious pair of feeding tentacles in many squids. Long, thin filaments continue from each appendage and bear microscopic suckers along portions of their length. Delicate tissues are easily damaged by nets, explaining why complete filament lengths are rarely preserved. Deep water supports soft bodies through buoyancy, reducing the structural demands imposed by gravity on land. A filament can remain extended with little heavy tissue, especially when surrounding water carries much of its weight. This physical context makes extreme slenderness viable without requiring rigid internal support. Large fins can generate slow, controlled thrust and stabilize posture around the short mantle. Video records show individuals holding arms away from the body while filaments descend or trail. Some have hovered near the seafloor, whereas others were recorded in open midwater. These observations establish posture and habitat snapshots, not an entire daily routine. The family has been recorded across widely separated oceans, suggesting broad distribution in deep marine environments. However, sparse exploration makes true abundance and regional boundaries uncertain. Taxonomic work compares morphology and genetic material, but damaged specimens complicate species-level identification. Researchers can confidently discuss the family while remaining cautious about naming every filmed individual. The animals live below most sunlight, where visual contrast, bioluminescence, and water movement shape sensory opportunities. Their eyes are large enough to support vision in dim conditions, though exact visual performance remains unmeasured. Like other cephalopods, They possess flexible muscular appendages controlled without bones or joints. The apparent elbow is therefore a maintained bend, not a hinged skeletal structure. Muscle arrangement can hold the thicker arm portion, while the distal filament responds passively to current. That division of labor offers a plausible mechanical explanation for the famous angular pose. It does not yet explain what the filaments contact, detect, or capture. The verified anatomy makes function the sharper and more difficult question. One mainstream hypothesis proposes that extended filaments intercept small organisms drifting through surrounding water. In this model, current provides the cue by moving prey against sucker-bearing strands. Contact could trigger local gripping, followed by arm contraction and transfer toward the mouth. No complete feeding sequence has been filmed clearly enough to confirm that mechanism in the wild. Another possibility is active sweeping, with the squid moving slowly while filaments sample a broad volume. That strategy would trade speed for reach in a habitat where prey encounters can be infrequent. Observed hovering supports slow station keeping, but it does not distinguish hunting from resting or sensing. The animal sometimes holds appendages nearly perpendicular to its body before the filaments turn downward. This posture increases the spatial area occupied without requiring a large, energy-demanding mantle. You can interpret the resulting shape as a living net, but that remains a functional analogy. Microscopic suckers provide direct evidence for adhesion, not proof of prey size or capture frequency. Stomach contents from better preserved material could identify meals, yet available biological samples remain scarce. The filaments may also contain sensory structures, as cephalopod arms commonly combine touch and chemical detection. Specific sensitivity in bigfin squid requires direct anatomical and physiological testing. Vehicle lights introduce another limitation by altering the darkness in which normal behavior evolved. Thruster currents may bend filaments, shift posture, or prompt escape responses. A squid recorded beside equipment may therefore be real, but behaviorally disturbed. Perspective can exaggerate length when the body and filament occupy different distances from the camera. Laser scaling improves measurements only when both reference points and animal lie in a suitable plane. Without that geometry, precise length claims should be treated as estimates. Some videos show damaged or incomplete appendages, further confusing comparisons between individuals. Development creates another source of variation because juveniles may not share adult proportions or behavior. Known specimens cannot yet anchor every large silhouette to a described species. The family identity may be strong, while body size, sex, maturity, and species remain unknown. The impossible appearance thus grows from authentic specialization amplified by incomplete views. Extreme appendages probably let the squid interact with more water than its compact body alone could reach. That matters in deep habitats where meals may be sparse and pursuit energetically expensive. A drifting particle touches a filament, local suckers engage, muscular tension changes, and the squid gains information. Whether the final consequence is feeding, rejection, or withdrawal has not been directly resolved. When a vehicle approaches, light and pressure disturbance may cue fin movement and altered arm posture. The squid can then rotate or glide away, preserving distance while its filaments stream behind. Such observations document responses to machines, not necessarily responses to natural predators. Potential predators include deep-diving whales and large fishes, although specific encounters are seldom witnessed. Long appendages could increase detection risk while improving reach, creating an ecological trade-off. Filament loss might be survivable if tissues regenerate, but regeneration rates in Magnapinnids are not established. Because cephalopods often grow quickly, life history may be short, yet Magnapinnid longevity remains unknown. Reproduction is even less visible, with courtship, mating, egg placement, and hatchlings largely undocumented. These missing stages prevent confident population estimates or assessments of resilience. Deep mining, warming waters, and declining oxygen could alter habitats occupied by poorly known deep animals. Specific impacts on Bigfin squid cannot be quantified without baseline distribution and abundance. Their wide records show that the deep ocean connects distant regions while still hiding local patterns. Each careful observation adds behavior, depth, and context to a family represented by fragile fragments. If you watch the filaments sway, the important question becomes what changed immediately before that motion. Q-linked observations can separate passive drift, active positioning, disturbance response, and possible prey contact. Future cameras need calibrated scale, low light, and long stationary deployments near undisturbed deep habitats. Stereo imaging could reconstruct three-dimensional posture and correct misleading perspective. Recorded current direction could reveal whether filaments drift passively or move independently. Close imagery might resolve sucker contact without touching or collecting the animal. When collection is justified, pressure-aware handling could preserve delicate tissues for anatomy and genetics. Stomach analysis from naturally damaged specimens could test whether the diet contains drifting crustaceans or other prey. Repeated observations of the same action would be stronger than a single dramatic encounter. A confirmed feeding event should show contact, attachment, retrieval, and ingestion in one interpretable sequence. Sustained natural observations could reveal how long the angular posture can be maintained. Filament reactions could establish whether distal structures detect chemicals, touch, vibration, or several cues. Juvenile and adult specimens would show how the extraordinary proportions develop through growth. Better genetic references could connect remote images, collected tissues, and geographic records more securely. You would still see an uncanny animal, but its shape would rest on measurable mechanics. The decisive evidence will come from behavior observed before machinery changes the surrounding water. Until then, feeding models remain hypotheses constrained by real suckers, real muscles, and real currents. The family's rarity in collections reflects fragile anatomy and difficult access, not supernatural origin. Its long filaments obey the same fluid forces that shape every drifting organism in the deep. What looks impossible is a genuine squid whose unobserved actions, rather than its existence, remain the mystery.